thank you for this opportunity. I present some of the concepts of the AQUAIMPACT project. So AQUAIMPACT is it's an innovation action project. So we are very applied, and we develop methods to help the European aquaculture industry, and especially in this case the breeding industry. And I apologize for the title, that it's very ambitious, but you see the point at the end of the presentation. And I make the bridge from AQUAIMPACT to AQUA-FAANG, which is developing novel approaches and novel genomic tools to help breeding programs, to make the let's say the conceptual bridge between the two approaches. About the selective breeding, I show this always just to emphasize to everyone that what is the big picture with the selective breeding. So with selective breeding, we aim to make food production that is profitable, so it makes sense to practice this as a commercial activity, and that the production is more ethical and then more environmental friendly. And this is the big goal of all. And Sara already described about the breeding genomic selection approaches. I focus especially on the computational part, and the breeding values are used to find the genetically superior broodstock fish, and they can be superior for growth or disease resistance or product quality. And traditionally, this has been done using this sire-dam offspring pedigree, which is very categorical. And in genomic evaluation, we use tens of thousands of DNA markers, we call them SNP markers, that are distributed across the whole genome of the individuals. And using these markers, we calculate what we call realized genomic relationships between the individuals. So we get rid of this categorical sire-dam offspring, but rather we calculate that genomically how similar individuals are across the whole genome. And we use this information to identify that genomically which broodstock fish are the best ones. And the built-in assumption with this method is that all the markers that we use, they are able to capture most of the genetic variation, because we assume that there are many genes which have small effects on the phenotype. So this is called polygenetic mode of inheritance. And what is one power of this approach is that using this sort of logic, we can make very large computational models with tens and tens of different traits, and estimate the breeding values for animals. And this approach has been highly successful in breeding programs in livestock and in aquaculture. It's the routine standard currently. And in aquaculture, we have put quite a bit of emphasis on using the similar data, thousands of SNP markers, to scan the genome to find potential genomic regions that have large effects on the phenotype. So these are called QTLs, quantitative trait loci. And this table here is a small summary of some recent studies on the inheritance of disease resistance in aquaculture species. And they are sorted from the top to bottom based on this value, which is the percentage of genetic variance that is explained by just few SNP markers or this QTL. So at the top we have the famous IPN case. There's a next one is Vibrio. So in these disease resistant traits, up to 70–80% of the genetic variance is explained by a single genomic regions. And then at the bottom we have very polygenetic traits in which the mode of inheritance is that there are lots of small QTLs that each explain a very small proportion of the genetic variance. And this pattern that we find these large QTLs, it's very unusual. And the trend is that we see it in aquaculture species and for disease resistance. And of course, the consequence is that in the previous slides, we learned that the genomic evaluation with the GPLAP assumes that the mode of inheritance is more like a polygenetic mode of inheritance. So the assumption is violated. And then from the positive side, this means that we may have some extra potential now in the genome for disease resistance that we can take benefit out in several different ways. And in breeding programs, when we have these large data sets with extensive SNP data also, there are different computational methods how we can take benefit out of the fact that there are some genomic regions that have an unusually large effect on the disease resistance. The top three methods here, PSB, WEIGHT, genomic evaluation, and SNPLAP, what they all three have common is that they in a very direct way use all the data what is available in the breeding program, and they estimate specifically, or they model specifically the SNP marker effects on the phenotype, so that the assumption, they have an assumption that some of the markers can have these very large effects on the phenotype. And the breeders don't need to make any pre-assumption or manually try to identify the regions that are unusually active. But the methods themselves do it through the data. And this is very powerful approach because it helps to make it straightforward. The weakness of this method is that currently they are challenging to apply in a multi-trait approach, but there's development into this direction. Then the last one is the marker-assisted selection, and in this one we manually identify QTLs and markers that are associated with this genomic region, and if we want to use them in long term, we should validate that these markers actually are consistently, permanently involved in the determination, or they are linked to the determination of the phenotype. This marker-assisted approach, it has been actually mostly abandoned in livestock breeding because it's you need a lot of manual work, and this traditional genomic evaluation, when there are enough markers, then they typically are powerful enough to capture most of the variants themselves. But in aquaculture, this has become of interest because of especially the disease resistant has potential for this approach. All right, so in AQUAIMPACT and elsewhere, we have demonstrated that when we apply these methods or some of these methods, then we can take benefit of the unusual mode of inheritance for disease resistance, fillet color, some downgrading traits. And the result of applying these approaches is that we can increase the selection accuracy almost even to double it when we specifically model these traits. And why accuracy is important: if we are able to identify more accurately which rootstock fish is the genetically best one, then it obviously increases the genetic improvement by the same amount. Now the important thing, and this is what Luca already mentioned, is that with all these approaches, we don't need information about the genes themselves. The genes may be located under the QTLs, but in the breeding program, we don't need to know the genes. of course, the data what we produce in the breeding programs, like Luca showed, they can provide key information to identify the genes and then go forward with using that information. And next, I demonstrate this logic, that the breeding itself is fairly black box approach, but you can go further and identify genes. And I use this IPN story as an example. And of course, this is the most extreme QTL that has been found in aquaculture species. But this story really applies also to smaller QTLs or even very minor QTLs, because the logic is general. So the story goes so that 2008, 2009 in Scotland and Norway, they discovered that there is one specific region in the Atlantic salmon that determines almost completely if the fish are resistant or not against the IPN. And it was very serious, and it is a serious disease. And then it was only a handful of DNA markers that needed to be used to identify the genetically best broodstock fish. And the graph here shows, this is data from MOVI, that when they applied these markers in the broodstock, they were able to reduce the mortalities due to the IPN from millions to almost to zero, just in a few years. And at this point, it was not known what is the gene underneath the QTL. And then there were some more detailed genomic analyses, including fine mapping, and they suggested that it's the CDH1 gene that determines the IPN resistance. And this story was present for quite a long time. Then the gene editing technology was developed, and of course it was of interest to test with gene editing what is the causative gene. And then it was observed that no, it is not the CDH1 which is the causative mutation, but it was a gene next to it, this NED8 activating enzyme, which is the causative gene. And now this demonstrates now the issue that when we do selection, we didn't need to know the gene, or we could even think that it's completely different gene what it actually is. But then the approach is what, for example, in AQUA-FAANG and others are approaching, that when you go to finally to gene editing, you are really able to nail what gene is underneath the genomic determination. So these new technologies are helpful here. All right, then to summarize, so in the inheritance of disease resistance in aquaculture species, it's fairly unique in many cases, so that we have these genomic regions that have very strong effect on the phenotype. They have these major QTLs, and if someone can identify the gene, then major genes. We can take benefit out of that in breeding programs using specific statistical models, which are still under development also. And to make a successful breeding program, we don't need the information about the genes themselves. Our methods are able, the SNPs are able to capture the genetic variation what is there, and we can use them in selection. But the next step is that, and this is now the link to AQUA-FAANG, is that with the novel methods, we can understand more, we can get more detailed information about the inheritance of the traits. And this is what Sarah has, for example, has done. If you are able to identify the genes itself, this is a very rare occasion that it happens, but when it is possible, then we can use gene-assisted selection. And then an obvious path is to gene editing, that if we are able to identify a QTL, then some candidate genes, then the gene editing is a pathway to make this sort of even more targeted genetic changes in the populations. All right, that's all from me. Thank you.